So the process of DNA profiling, they take a sample of DNA and they put it into a gel plate, and then they use electricity, and the portions of DNA separate, and it makes a unique sequence of bars or bands. So here's an example of DNA profiling. We had a victim, and at the crime scene, they found some sample of DNA. So maybe on the handle of the knife, they found that there was some skin that was on there. It's important that they get a DNA sample of the victim and they compare it with the sample found on the crime scene. So if the skin on the handle matched that of the victim, we know that that skin also came from the victim. So it's no use looking for another suspect there. But in this case, we can see the sample at the crime scene does not match the victim. The first bar matches, but from there on, we find that there's a lot of differences there. Right, so if we look here, we find that this first bar matches exactly with suspect 2, the second bar as well, the third bar in the same line, the fourth bar, the same position and the same thickness as well, the fifth bar, as well as the sixth bar. Right, so generally in a question paper, you take a ruler and you'll verify that each of these are in the correct position. So in this case, scientist or the investigator would have proven that the sample found at the crime scene was from suspect number 2.